What?
All right. So today we are going to pick up where we left off before the exam and finish stuff out. Exams looked okay. Average was like a high C. So a little bit worse than our last two exams. So let's make sure to keep up on our study skills. I know it's getting towards break. Hoping having a little less content in that unit would be better, but that's fine. We'll recover. Okay. Yeah, and a reminder, my office hours are tomorrow most of the day. We do have candidates interviewing in the chemistry department, so I'll have a couple more meetings than I usually do. But my Friday afternoons are also open, so if you want to come and talk to me about your exams immediately, we can do it this week, or I should have some more time after this week, although I know our break is coming up. Okay, so back to skeletal muscle. Okay, so when we left off, we've been talking about the twitch, and in addition to learning the general parts of the twitch, we had mentioned that we have these two types of twitches, right? So we have our isometric and isotonic twitches. So I just wanted to put in a little example here for us, because for those of you moving into kind of clinical practice, you might be working with different types of exercises and thinking about these different types of twitches. Take a look before we review. Okay, great. Yeah, so in this example, we're looking at therapy after a quadriceps injury. So that big quad group in your anterior thigh, right? And one of the things we might be doing is asking a patient to push against a resistance bar that is immovable, that they can't move, right? So we would expect to see the muscle contracting, right? That's part of it. We're trying to build strength. But we wouldn't see the overall length of this muscle changing because we're not actually creating movement in terms of moving a load. So this is an example of like an isolated isometric twist, an isometric muscle contraction, right? Where we see our tension building, right? So we have our latent period, we have tension building during the contraction, and then we have relaxation as our tension tapers off, right? And so we'd see all those phases of the twitch if we were measuring tension from that muscle belly, but we can see that our overall length of the muscle is staying the same. And that's different than our isotonic muscle contraction, where we actually move a load. So our graph of tension would look a little different because in an isotonic muscle contraction, once we are able to actually move the thing that we're pushing or pulling on, we are going to see our muscle overall shortening and the tension kind of flattening out as you try to put just enough force in order to move the object that you're trying to move. So we see shortening and a leveling out of tension in an isotonic twitch. Okay. So now we're going to be thinking about how we have different amounts of strength, different amounts of force from a muscle. Okay, so that's our graded muscle contraction. So small, all the way up to large amounts of contraction. So we're looking at how much tension a fiber produces, an individual muscle fiber, And we're looking at the whole muscle level, how many of those fibers are contracting at any given time. Okay? So when we think about it at the fiber level, we're thinking about how many cross bridges did we form as we were contracting our sarcomeres, right? So as we were doing that our sliding filament model, right? We know that we're doing the cross bridge cycle, grabbing onto the actin with our myosin, pulling it in with a power stroke to make that sarcomere shorter. So if you form more cross bridges, you will have more force because that sarcomere gets shorter. But you can also increase strength, increase contraction by having multiple muscle fibers contracting at once. So when we look at tension in a muscle overall, one of the reasons we do warm-ups is so that we limber ourselves up, but also so that we get to the point where we are actually able to use our maximum tension, our maximum force. Okay. So what we're looking at here is what happens when you have multiple stimuli and you contract your muscle multiple times in a row. Right, so one thing that we can notice, in particular on this graph, is that we've got this kind of staircase. Right, so we've got our first muscle contraction, a little bit of tension, our second, a little bit more, and so on until we hit this plateau. So this is the maximum tension that we're able to generate from that muscle or muscle fiber. So here's our warm-up, and then we're on with the game as we get to this plateau. So that staircase is checked. The other thing that we can see with successive contractions is this thing called summation. Okay. So when we think about why we see summation, when we think about what summation is, we want to remember that our contraction fundamentally is a response to an action potential. Right? So we have our motor neuron coming to the neuromuscular junction, ultimately creating an action potential in the muscle itself. Right, so an action potential is going to last like 2 milliseconds or so, and our contraction is going to depend a lot on specifically what muscle fiber we're talking to, what muscle we're talking to. But contraction is going to be definitely longer than an action potential, right? It's going to be smallest, maybe 10 milliseconds, but up to like 200 milliseconds is a reasonable length of a contraction as we're forming those cross bridges, shrinking our sarcomeres by pulling in actin. Okay. So this means if you have a 2 millisecond action potential come in, and then you contract for 10 seconds, or sorry, 10 milliseconds, 10 seconds would be a lot more, right? But you still have space in that 10 milliseconds for another action potential to come along. Right? We remember that action potential never change in size when we're thinking about neurons, but we can, change their frequency so long as we keep in mind that they have to be spaced out by a refractory period. But when we think about our contractions in muscle, there isn't the same refractory period as when we looked at action potentials in neurons. So I'll show you what I mean. Okay. Right. So here we have our graph of a twitch laid onto an action potential, right? That stimulus and then that signaling in our excitation contraction coupling, getting from the actual signal from a neuron to you actually doing the cross-bridge cycling, right? So here we see the reaction to the action potential, short action potential, and then a long time while we go through the phases of the twitch. Okay. So if we have frequent action potentials, we're going to see that we can have our twitches kind of stacked together. So this is a little bit like when we talked about graded potentials in our neurons, right? So a single twitch is a response to a single stimulus, right? But if you have multiple twitches in a row, if you don't give enough time for the muscle to fully relax in between, like we saw in the first graph with the trepe, we saw full relaxation between our twitches. But if you don't have enough time, right, and you hit the muscle with a new stimulus, while it's still not quite done relaxing, it's just going to twitch from that point that it was at, right? So we're going to stack on our next switch, and then our next switch, right? So we see this stair-stepping effect again, right? So kind of like Treppe, but what we're focusing with the summation is that the bottom of this trough, right, is getting higher and higher because these action potentials are coming in quickly, right? So this can happen even faster. So here we see that we've warmed ourselves up with this staircase Treppe. We can see summation is happening because we're not fully relaxing. And we're hitting that plateau of our like maximum amount of tension that the muscle can produce, right? So we're getting towards what we refer to as tetanus, okay? So tetanus, complete or fused tetanus, is when you are getting up to the maximum force that you can, the maximum tension with this muscle, and you are holding that. And so no matter how many more action potentials you have come in, no matter kind of how fast they come in, right? Once you've hit this maximum tension, all we can do is hold it flat. So we would see if you're in complete tetanus, right? We no longer see any relaxation at all between our action potentials coming in and our twitches forming. We see this solid continuation of that maximum tension. Here, this is labeled incomplete tetanus or unfused because we get up to that maximum tension and we see multiple spikes in a row to that maximum tension, but we still were trying to relax a little bit in between. So that's the only difference between those there. So one of the factors then that's going to affect our force generated by muscles is like, did you warm up? Did we have this Treppe, this kind of stair-stepping effect of our tension generation? And then did we have summation, right? Did we have these sequential action potentials adding together to get you up to high tension? But we can also look at the fibers themselves to understand our force being generated. So any muscle in your body is going to have an inherent amount of force that it can generate, right? So we've got a maximum, right? And what that maximum is going to be dependent on some of the properties of the fibers. Okay. So one thing, we know that the contractile part of the muscle is really those contractile proteins, myosin and actin, so thick filaments and thin filaments, sliding past each other to make sarcomeres shorter, right? So one of the things that's going to make a muscle more forceful is if you have more sarcomeres. Specifically, these sarcomeres actually have to be in parallel because there's a certain amount of space a sarcomere takes up, right? So the way we're going to be able to stack more is actually kind of next to each other rather than more end to end. Okay. The other thing that's going to be able to get us to make more force is if we have more cross bridges in general, right? So like just more calcium, more cross bridges, shrinking these sarcomeres shorter. Okay. This means there's a relationship between the diameter of a muscle fiber and how much force it can generate, right? So if you think of a thick muscle fiber, right, like a big one, right, you're going to be able in that cross section to fit sarcomere, 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 right? The bigger diameter to a muscle fiber, intrinsically it's going to be able to generate more force because we have more of these sarcomeres and we have more cross bridges being able to form, in part because we have more sarcomeres. Okay. So when we see large diameter muscles and muscle fibers, they're going to be stronger. They're going to generate more force than when we have small, skinny ones. Okay. So when we think about, say, an individual muscle fiber or an individual muscle, right? Sometimes your muscles are going to feel like they're weaker and they can't move loads as well in certain positions as in others. So part of this is physics about like how you have to lift that load, but part of this is actually also about how stretched out that muscle is. So our muscle fibers and our sarcomeres actually have an optimal length, right? Like, so they have a perfect starting size where they're able to generate the most force, right? So if we are either too stretched out or too bunched together, it becomes more difficult to generate force. And like the maximum force that you can generate from that muscle is going to be a little less. So our perfect length is basically when we have all of those myosin heads overlapping with some actin, right? But we don't have any of those myosin heads, those cross bridges, like in the middle, in that the part where the actin isn't, and we don't have them like bunching up at the Z line. I'll show you a picture. So fortunately, your muscles in their relaxed states should be approximately at their optimal length, right? We want to be able to use our muscles efficiently and effectively, but either of our situations that are not optimal are going to make our muscles a little weaker, a little less useful. So if we have a longer sarcomere, a kind of stretched out one, we have not really enough overlap for our cross bridges. And if we have a short one bunched up, we're going to have our thin filaments even kind of overlapping each other. I'll take a look at a second. All right. This kind of goes with the summation and tetanus thing before we take a look at that graph, kind of pulling everything together. So in case you were curious about why we see summation, why we see tetanus, that has to do with the calcium. So basically relaxation, that needs to clear out the calcium from the cytoplasm of the cell so that we stop cross-bridge cycling. Right? So if you have lots of stimuli in a row, some of that calcium hasn't been cleared yet. Some of it's still sticking around, which means our cross-bridge cycling keeps going. And then when we hit tetanus, we've got our troponin completely full with calcium. So there's basically no more cross bridges you can form. You're at the maximum number. Okay. So here's that optimal length thing. So what we're looking at here is we can see the sort of like normal length of our sarcomeres. We're going to think of as this or this, approximately, right? We've been drawing them specifically like this a lot, right? With that bare zone, kind of wide, relaxed state, right? And then contracting that way. Okay. But here we have what happens if we have either too short a sarcomere or too long a sarcomere. Okay. So let's start with the long one. Do stretched out muscles, right? So if you're extra stretched out, you're kind of at the limit of how long that muscle can get. What we can see, right, is that we've got some of those cross bridges, some of those myosin heads that don't have any actin to grab onto, right? So this means that when they start contracting, right, even if we have enough calcium, even if we have the right action potentials, right, this is going to be harder to actually generate force with because only the ones that have neighboring actin to grab onto are actually going to be able to pull that actin in towards the center. So it's harder to get started when you're at this longer stretched out length, and you're weaker at this longer stretched out length because you have to shrink the sarcomere back to a good amount of overlap in order to get full use of that muscle fiber. So here we can see that even more extreme here for C. So here we can see that the maximum tension we could generate from this sarcomere is basically zero, because even when we have calcium uncovering those myosin binding sites on the actin, right, there's only maybe one, maybe none, if we're actually at zero, places where we can grab together. Right, so this is why too stretched out a position is a weak position for you. On the opposite side, we don't have that same problem, right? We've got enough actin to grab onto, right? So we've got myosin and actin overlapping everywhere. But we'll notice that we don't really have any gap here, right? And also, if we look at that thin filament, right, we now see that the thin filament is actually overlapping itself in the middle of the sarcomere. So at this point, we also have a decrease in how strong that sarcomere is going to be able to contract, right? Because it just doesn't have enough room, right? It can't get much smaller, right? Because to get smaller, you're going to have to now kind of like crinkle things up, right? You're basically blocking yourself with the other half of the sarcomere. So when we say we have an optimal length, this is what we mean. We want the perfect amount of overlap between those myosin heads that form our cross bridges, but we want enough space to actually be able to shrink this without running into other structures. So now we're going to think about whole muscles rather than just muscle fibers. We're going to link this together a little bit. So more sarcomeres contracting means more force. Also, more muscle fibers contracting means more force, right? So just like stacking on how many structures are actually pulling at the same time makes it easier to move stuff, right? So we have this concept of recruitment. Okay. So in recruitment, basically what we're doing is we're going to ramp up how many muscle fibers are contracting in order to meet the demands of the exercise that we're trying to do. So our big muscle is just a bunch of muscle fibers, so bundles together. We know they're grouped into motor units, right? And so we are going to grab more and more motor units, bigger and bigger motor units as we try to make stronger movements. Okay, so we're going to take a peek again at our motor units. Okay, so here we're looking at two different motor units, motor unit X and motor unit Y. We remember that a motor unit is a motor neuron, but then splits and links up to multiple muscle fibers, right? So any given muscle fiber goes back to one motor unit, not multiple, one motor neuron, right? But any given motor neuron goes out to multiple muscle fibers. Okay. So we are going to see that we have motor units of different sizes, so connected to different numbers of fibers and different sizes of fibers as well. So we'll have small little motor units, say like 5 fibers, then we'll have larger motor units, and we can get the most force if we contract multiple motor units together at the same time. Okay. So what's happening when we do motor unit recruitment is that we are trying to contract more muscle fibers, right? But we're a little bit lazy. We don't want to waste our energy. So we don't just contract every single fiber in our muscle at once, right? That would waste a lot of ATP. Okay. So what we're going to be doing is we're going to be activating our motor neurons sequentially, right? We're going to be activating our smaller motor units before we activate the larger ones. Okay. So every time we activate a motor neuron, activates the entire motor unit, okay? And then we see increases in how much force comes from a motor unit, depending on how many fibers we have and how big they are. So when we are trying to make delicate movements, little precise movements, right, you can feel that doesn't take a lot of force. Like you're hopefully not gripping your pencil really hard, maximum amount of force that you can use while you're trying to write, right? You're holding it with some pressure, but relatively loosely, right? You want to be able to do that for a long time. You want to be able to do that kind of precisely. And these are relatively small muscles. Versus if you're trying to lift weights or do push-ups, right? You'll feel that that's a much greater amount of tension you're building. You have much larger muscles that you're using, and we would see that we are using larger motor units. So more muscle fibers, more cross bridges, more sarcomeres, large groups of fibers together. So we see that any given Moon's muscle is going to have different numbers of motor units within it, right? So that's one thing that can vary. Okay. And it's going to have motor units that vary in size. So we'll have within, like your biceps, you'll have some small motor units, you'll have some medium-sized motor units, some larger-sized motor units. Okay? In general, when we are looking at like an eye muscle versus your quads, right? Like our smaller muscles, things that do more delicate movements are going to have more of these small motor units. Okay. Within a motor unit, we're going to see that those small motor units for delicate movements also tend to have smaller fibers, right? So they're going to be a little weaker. And those large motor units that are adapted for strength are going to have larger muscle fibers within them. Okay. So imagine what type of fiber, what type of motor unit you're going to expect to see if you just did a surgery and you're about to suture your patient close. So you're putting in the stitches. Right, so we're trying to thread our needle. What kind of motor unit are we going to be using there with our hand? Okay. Yeah. So we're starting out with a small motor unit as we're just trying to put thread through the eye of a needle. But once you're actually trying to push that needle through the skin to actually make your sutures, you're going to be using slightly larger motor units, right? Because you have a variable amount of force that you want to create in that muscle. So this leads us to the size principle, right? So we are going to ramp up how big of a motor unit we are recruiting in order as we try to use more force. So we use our small units first, and then we use our large units last. Part of this is also a byproduct of how we create action potential. Our large motor units, so lots of fibers that have big diameters, also kind of need larger neurons to go to them because we have to split it out a bunch at the end to more fibers. And when we have a larger neuron, it takes a little more time. It's a little more difficult to depolarize it to threshold, right? Because you have to have more channels open to let more sodium in. It's just going to take a bit more to actually be able to depolarize the large neuron and then these large motor units. So this kind of requires like more signaling from our synapses in the central nervous system before we send signals out to our periphery on these motor neurons in order to actually recruit these larger motor units. So this is helpful for us in that we like to only recruit the amount of muscle that we need for a given action, but it also has kind of a practical constraint on it. So 2 reasons we see this. Okay. So what we're saying in picture form is just this, right? So all of our motor neurons are getting some kind of signal from the brain, right? Like you thought, hey, I need to lift up this water glass or something as you're taking a drink, right? So you have this thought from your brain. Your brain says, great, I'm going to go send out the signal, right? So that's coming down your spinal cord. So all of this is central nervous system stuff. So stuff we did in unit 2. Okay. What we talked about in Unit 3 just now is the somatic nervous system part, right? So going out to the peripheral nervous system. So here are our 3 motor neurons. So here we have motor neuron X. Here we have motor neuron Y. And then motor neuron Z. And we can see that they're getting larger, right? X is only attached to 3 little muscle fibers. And by the time we get to Z, we got 1, 2, 3, 4, 5, 6, 7. Okay. So what we would see in terms of ramping up our tension through ramping up the recruitment of larger motor units is we'd see that first we recruit motor neuron X. It's relatively easy to depolarize because it's a little bit smaller. So that signal gets out to these motor fibers, these muscle fibers faster, right? But they're not that strong because they're kind of small and there aren't too many of them. So we only produce a little tension when we recruit that motor unit. But we haven't hit the amount of tension we need to, what are we doing? We're lifting a water glass, right? Okay. So we're going to keep signaling, right? And now, after a little bit of time, we've got enough neurotransmitters coming to this synapse with our motor neuron that we're able to depolarize motor neuron Y, right? So when it depolarizes and sends its action potential, it grabs more of our muscle fibers, right? So now we've got 5 medium-sized muscle fibers contracting. The muscle fibers from this motor unit X, they're still going, right? So they're not relaxing yet. So we can see that just like in our summation graphs, this is an additive effect, right? So we're getting a little more of a boost from Y stacked on top of our tension that we produce with X. Same thing happens when we get to Z. It takes more time because this takes even more to depolarize. But once we do, we see a big jump in how much tension we're producing. And that's how we get to our maximum contraction from this larger whole muscle with multiple motor units in it. Okay. So we are now going to switch over to intro our skeletal muscle metabolism. Okay. So our skeletal muscle metabolism packet will be grouped into Unit 4. So we'll be doing muscle metabolism and cardiac for our final, for exam 4, I should say. Okay. So as we're getting started with this, I want us to review metabolism itself briefly. So go ahead and join us as we walk through a couple of questions, jogging our memory on stuff we've seen before that's going to be related to this concept. So starting ourselves off, let's try and remember what muscle we're oxidizing during aerobic cellular respiration. So we did this in our metabolism unit. Okay. Yeah, great. So our big goal in cellular respiration, the main part of metabolism that we focus on, is to oxidize glucose. It's a glucose oxidation process where we're breaking up glucose, pulling the energy out of glucose, putting it into electron carriers that we then use to fuel our ATP production at the end, okay? So this is a series of reduction oxidation reactions, but what we're doing overall is oxidizing the glucose. Okay. So let's also remember storing glucose in an animal cell. Right? So what's our molecule? It's basically a bunch of glucose linked together. Okay, yeah, it's glycogen. So glucagon was one of our hormones for our endocrine stuff as we were thinking about absorptive, post-absorptive, right? Our hormone of the post-absorptive state. But glycogen is our storage molecule for extra glucose, right? So we make glycogen when we have excess nutrients, excess glucose coming in when we're eating, and then we can rip apart our glycogen later on. So thinking about our cellular respiration and our glucose oxidation, what was the step where we're making lots of ATP? So all of the steps on the slide here can make ATP, some ATP, but remember glycolysis on net only gets us to ATP. The Krebs cycle, we got one per turn of the cycle. So again, just two in terms of per glucose molecule. And it's the oxidative phosphorylation at the end. So after our electron transport chain and the acceptance of that last electron by oxygen, that we have our hydrogen atoms or protons flowing through ATP synthase into the mitochondrial matrix, making us on the order of 30-ish ATP per molecule of glucose. So it's this oxidative phosphorylation step that's the big one. Okay, so let's remember, we don't have oxygen. How would we be making our ATP? Because we can still make some ATP when we don't have oxygen. We got some diversity of viewpoints here. Okay, so what we want to remember, when we don't have oxidation, or sorry, oxygen, everything inside of the mitochondria gets kind of backed up, right? We get jammed because we don't have like the turnover of our electron carriers, right? So we make a bunch of reduced electron carriers. We make the NADH, right? But we can't get back to NAD plus without the oxygen at the end of that electron transport chain. So we stop doing our oxidative phosphorylation step, that part where we're flowing with a lot of protons across that inner mitochondrial membrane, but we also stop the Krebs cycle from turning because we run out of NAD plus, which is really what the Krebs cycle, that TCA cycle, is all about. It's all about making our reduced electron carriers to send to the electron transport chain. So what we want to do in a low oxygen environment, or in the complete absence of oxygen, is we want to ignore our mitochondria completely. We want to stay out in the cytoplasm, and that's where glycolysis happens. So when we have no oxygen, we're just going to do glycolysis over and over and over again, which is why we have that lactic acid fermentation step, right? So where we turn pyruvate into lactate, right, in order to regenerate the NAD plus that we need in order to do glycolysis over and over again in the cytoplasm. Okay, great. Right. So the reason we're doing that is because we are going to see that when we think about muscle metabolism and we think about different types of muscle fibers from this metabolism perspective, we're going to see that different muscle fibers are specialized to do a lot of oxidative phosphorylation with a lot of mitochondria, a lot of oxygen. And we're going to have muscle fibers that are specialized to not need so much oxygen, to be able to work in low or no oxygen environments and just do that glycolysis stuff over and over again. It's going to be one of the main divisions between our types of muscle fibers. Okay. So going back to how our muscle cells are making ATP, right? Overall, we need ATP in order to do work, right? To generate that tension that we were talking about. We needed to do the cross-bridge cycling. We know that in the cross-bridge cycle, right, at the end or the latter half of the cross-bridge cycle, we're grabbing ATP molecules, pulling them apart in order to reset ourselves in a position such that we can grab another actin and do the power stroke again. And so we need ATP in order to contract a muscle. Okay. And we can make ATP in a couple different ways, right? So the two main ones that we think of, right, are so far numbers 2 and 3 here, right? So either our oxidative phosphorylation using our O2, and we just talked about glycolysis, right? So happening way out in the cytoplasm early in our process, which we can still do without mitochondria if we take this lactate pathway. Okay. One thing we haven't talked about yet, because this is going to be more specific to muscle fibers rather than just general ATP, is this creatine phosphate ADP kind of system down here. Okay. So what we can do is we can phosphorylate, so add on a phosphate group to ADP, our adenosine diphosphate, right? So our precursor molecule for ATP, basically. Right? Using this creatine phosphate, which is going to give us ATP, the creatine part of the creatine phosphate left. Right? So this is what you're kind of trying to do if you take creatine before you go to the gym or something, right? You're trying to get more of this step kind of going to get a little extra energy. Okay. So you should have this to some extent already in your muscle fibers. So at rest, you've got a little bit of ATP, and you've got some creatine, and you've got some creatine phosphate in your cells. Now, doing what we're doing in this equation with this creatine phosphate system is faster than glycolysis and faster than oxidative phosphorylation. So we're going to think about this as being like almost immediate energy, just like on tap, ready for your muscle fibers to use, right? So what's happening, right? We just said we take creatine phosphate, we grab that phosphate group, add it to ADP, which means that we're left with creatine and ATP here, right? So we have an enzyme that's responsible for this. So here's our creatine kinase, right? And this reaction can go forward or backwards, right? So if we think about how reversible reactions work, right? So we can see that this isn't just a one-way arrow, right? So it goes both directions, right? What we're going to see when we use the system is that this is going to operate by the law of mass action. So basically, whatever you have more of, like whichever side is bigger, either this left side or this right side, we're going to go from the side where we have more of our reactants towards the side where we have less. Okay. And vice versa, right? So if we had lots of ATP unused, right, this reaction might tend to proceed the other way. So the way that this is getting driven, right, is that as you start contracting your muscle, as you start exercising, you're exhausting this ATP, right? So you use up this ATP, right? So you're dropping down how much ATP is left, right? So we push this reaction to the right by the law of mass action to make up for that. So to replace all that ATP you're using. Okay. So this works to get you about like 5 times the amount of ATP that you have at rest. So this can't go on forever. You know, it's in limited supply, but it's going to be really fast. So we're going to see that when you start exercising, actually kind of regardless of which type of exercise you're doing, right, we're going to be using this creatine phosphate system to jumpstart our exercise. Right, so here we're looking at the energy and where it's coming from for our exercise. So here you see that creatine phosphate system is our first kind of thing that we're. Now, depending on what type of exercise you're doing, whether it's like light or kind of moderate, right? So we're thinking like maybe some yoga, something slightly lower energy, right? We're going to have a different graph of where that energy, that ATP is coming from than if we do a really high intensity exercise. Okay. So we're going to use creatine phosphate, our anaerobic glycolysis, and our oxidative phosphorylation in all of them, but they're going to be in different amounts. And we're going to see, one of them predominating over the others in each type of exercise. Okay. So we start with our creatine phosphate, okay, because that's easy, that's quick. But what we're ultimately going to get to as you do a light to moderate intensity exercise over time, right? Once you've been doing that exercise for a while and you keep going, we're ultimately going to shift our focus to our oxidative phosphorylation, right? So like once we're warmed up and we've been doing this exercise for a little bit, what our muscles are going to be trying to use is our energetically efficient way of producing ATP. We're going to be, trying to use oxygen to make lots and lots of ATP as we go further. Okay. So first we start out making ATP with this creatine phosphate system. And then we get our glucose oxidation going, right? So we start breaking up glucose. And at first, that glucose is going to come from our glycogen stores that are in the muscle, okay? So it can take kind of a long time to pull glucose or fatty acid out of the blood while you're starting to exercise. So it is important to have some extra glucose available to you, but it's going to take a minute to get into these cells. Because we're relying on oxidative phosphorylation at the end, the key thing that we're going to make sure we know about these light and moderate intensity activities is we need oxygen. So you need to be breathing. You need to be breathing potentially a lot, right? Because you need to be getting lots of oxygen in. You need to be pumping it out to the muscle, and that muscle needs to get it. So you want those vessels to be nice and big and easy for blood to flow through. So we'll pick up here next time and compare that to what happens when you're trying to use bigger, stronger muscles to do higher intensity activities.